Okay, hello everyone. Welcome to PION's webinar entitled The Impact of Colloids and Nanoparticles on Intestinal Drug Absorption. That is part of our monthly fiber optic user group meeting as well. I'm Balin Shinko with PION and I will be the moderator of this event. Today we will have two presentations on the topic, one by Professor Prof Sugano and one by Dr. Nali. We want this webinar to be interactive and therefore encourage you to contribute to its success by asking questions at any time during the presentation through the Q&A box on your screen. Any questions that we don't get to today will be answered by email after the event. If you need any technical assistance during the webinar, then please type your query into the chat box and send it to the moderator. Also, a recorded version of this webinar will be available on our website, and an email with the link to the recording will be sent to all registrants as well. The first presentation is an overview of the particle drifting effect by Prof Sugano. And just stop sharing my screen so he can take over. And Prof Sugano is a professor at American University. He has over 20 years of experience working within the pharmaceutical sciences in Japan and in the UK. He received his bachelor's and master's degree in chemistry from Waseda University and his PhD in pharmaceutical sciences from Toho University. His research interests include drug oral bioavailability, where he has established the gastrointestinal unified theoretical framework that is a widely accepted and commonly used mechanistic modeling system for oral drug absorption. In today's presentation, he will introduce the concept of particle drifting effect and its potential impact on drug absorption. And I can see he already shared his screen. So, Professor Sugano, please start your presentation.
Okay, thank you very much. Okay, I think you are seeing my slides. So, thank you very much for your kind introduction, Balin. So, I'm Prof Sugano. Please call me Kyo. I'm now working at American University in Japan. It's 11 o'clock in the evening, so now actually it's good evening for everybody. So the title of my presentation is Overview of Particle Drifting Effect. So this slide shows the outline of my presentation. First, I'd like to talk about the theoretical FA is a fraction of those absorbed. So FA and PEF equation. PEF is effective permeability across the intestinal membrane. And then move on to the real drug discovery and clinical study data, which suggest the existence of particle drifting effect. And finally, I'd like to talk about the microflux to FA extrapolation, considering the particle drifting effect using the predictor. So this slide shows the concept of oral drug absorption. When we administer a drug product, the disintegration process is usually very fast. So basically, the drug substance is released from the formulation, and the drug substance then dissolves into the intestinal fluid, and the dissolved drug molecules permeate the intestinal membrane. So the dissolution process is usually described by the Noyce-Whitney equation, and the permeation process can be described by the first order kinetic. Therefore, so we can calculate the fraction over dose above, fraction over dose by integrating these two equations. So this slide shows a quest for the equation. So basically, we have a very long history about for the equation. The Noyce-Whitney equation was proposed in 1897, and 100 years later, in 1985, the absorption potential was introduced. And then 1993, the plug flow model was introduced by Professor Amidon's group. And in 1995, the BCS concept was introduced. And around the same time, maximum absorbed dose and compartment absorption transit model were introduced. And in 1999, absorption limiting step classification was proposed. And in 2009, FA equation was finally found. And in 2010, based on this FA equation, FA classification system was proposed. It is now called FARLS is Sparrows equation classification system. So this slide shows the concept of absorption using the three-bucket model. So this first case is called dissolution rate limited absorption. In this case, the dissolution rate is very slow, but the permeation rate is very fast. So in this case, the oral absorption is determined by the dissolution rate. So this case is called dissolution rate limited absorption. The second case is called permeability limited absorption. In this case, dissolution process is really fast, but the permeation process is very slow. In this case, the drug molecule dissolved in the intestinal fluid actually accumulates here and slowly absorbed into the body. So this case is called permeability limited absorption. The third one is called solubility permeability limited absorption. This case is most important for current drug discovery and development. The maximum drug amount which can be dissolved in the intestinal fluid is determined by the solubility of a drug. In this case, the solubility means both that of a crystalline drug or amorphous drug. So when we dose a solid dispersion formulation, the maximum drug concentration is determined by the liquid-liquid phase separation concentration. When we dose a crystalline drug, the solubility is determined by the drug concentration is determined by the crystalline solubility. So anyway, in this case, the oral drug absorption is determined by the solubility multiplied by the permeability. So this part, so the permeability part, is very important to understand the particle drifting effect. So this slide shows about the concept of PEF equation. So after the drug molecules were dissolved in the bulk phase, they then permeate across the unstirred water layer, which is on the surface of intestinal wall. The thickness of the unstirred water layer is about 300 micrometer. And then the drug molecules permeate through the epithelial membrane. So this process, basically the drug permeation is across the intestinal wall, is a sequential process permeating through the unstirred water layer and then the epithelial membrane. And so mathematically, this process can be written by this equation. So here the effective permeability, and this part is the unstirred water layer permeability, and this part is the epithelial membrane permeability. Fu is the free fraction in the intestinal fluid because intestinal fluid contains some bile cells, and only the free fraction can permeate across the epithelial membrane. So this equation is called PEF equation. It's very important when we understand the oral absorption of a drug. So by using the FA equation here and the PEF equation here, we can derive this kind of decision tree to understand the rate limiting step for oral drug absorption. The FA equation contains a concept of BCS. So BCS is, basically the classification system using the dose number and permeation number. So basically, the FA equation includes the concept of BCS. So when the dissolution rate is very slow, it becomes a dissolution rate limited absorption. But in the case of permeability limited absorption, it can be further divided to the epithelial membrane limited case and unstored water layer limited case. So similarly, in the case of solubility permeability limited absorption, it can be divided to the solubility epithelial membrane permeability absorption and solubility unstored water layer permeability limited absorption. And this last case is where the particle drifting effect becomes important. So now let's move on to the real drug discovery and clinical data. So I wrote this paper in 2010. I think this is the first paper about the particle drifting effect. And at that time, I was working for Pfizer. So the particle drifting effect was actually proposed based on my real experience in real drug discovery. So in drug discovery and drug development, we often perform the dose titration study. So this slide, in this figure, the horizontal axis is the dose strength, and the vertical axis is the dose absorbed into the body. So theoretically, when we increase the dose, the observed dose hits the ceiling here. So this value is called maximum absorbable dose. So theoretically, the absorbed dose will not increase above this value. But in reality, we often observe this kind of increase in the absorbed dose. And when we reduce a particle size, the absorbed dose further increases like this. So especially, I noticed this when I saw the TK, the toxicokinetics data, or FIH study data at high doses. So, so this is a theoretical line without particle drifting effect, and these are the real observation. So we might be missing something in the theoretical equations. So this slide shows the very famous saying by the Sherlock Holmes. So when you have eliminated all which is impossible, then whatever remains and whatever improbable must be the truth. So this is a very famous saying by the Sherlock Holmes. So in the case of solubility limited absorption, the absorption flux is determined by solubility multiplied by permeability. And in vivo, in reality, the flux is increased as we increase the dose strength and reduce the particle size. Only for solubility unstored water layer limited case, but not for the solubility epithelial membrane limited case. So this point is very important. And the dose strength and the particle size do not affect solubility. We will discuss this point later. Therefore, in reality, when we increase the dose strength and reduce the particle size, the absorption flux increases, but the solubility remains the same. So the conclusion is that unstored water layer permeability should have been increased. So the next question is how the increase of dose strength and reduction of particle size can increase the unstored water layer permeability. So the particle drifting effect. So in the previous theory, we implicitly assume that the drug particle only exists in the bulk phase, but drug particles can actually penetrate into the unstored water layer. In this case, the mass transport rate across the unstored water layer can be increased. So this is the basic concept of unstored water. Sorry, this is the basic concept for particle drifting effect. And then so I looked into the literature for the evidence supporting the particle drifting effect. And actually, many articles suggested that the microscale particles can penetrate into the unstored water layer. So there's many papers showing this kind of data. So this is just a typical one. So as you can see that much portion of the particles exist in the intervellous space. So this is a clear evidence that microscale particles can penetrate into the unstored water layer. So this slide shows snow drifting. So actually, I named the particle drifting effect after the snow drifting phenomenon. So when a blizzard occurs, strong winds blow the snow up into the point where the wind is stagnant. So at this moment, I'm not sure whether the particle can be brought into the unstored water layer by the passive diffusion, so it's a Brownian movement, or by the convection, is by the fluid flow. I don't know which one is correct. Maybe both working, but I named that particle drifting effect, imagining this kind of phenomena for snow drifting. And so this slide shows the FA prediction by the FA equation. And the horizontal axis is a clinical FA percent, so fraction over dose, observed in humans. And the vertical axis is a predicted one based on these physical-chemical properties. It's pKa, intrinsic solubility, the biomicelle binding partition coefficient, and intrinsic passive transcellular permeability. So this P trans 0 value was predicted from the log P of compounds. Without particle drifting effect, there's many under predictions. And when we looked into these data, these cases are high dose and small particle, fine particle cases. So when we adding the particle drifting effect, the predictability was much improved. And so I published this paper in 2010, 11, sorry. And so basically, after 2011, I collected a lot of additional clinical FA data and predicted and plotted them against the predicted ones. And basically, the predictability remains the same. So now I'm really confident about the FA equation and the PEF equation. But this is not for the supersaturation cases. This is a simple non-supersaturation cases. So supersaturation cases, it is much more difficult to predict. So supersaturation cases include the solid dispersion formulations and salt form API and amorphous API and other cases. So the supersaturation case is much more difficult to predict. And that now we are trying to predict based on the in vitro data, but it's still very difficult. So this slide shows the effect of particle size on solubility. The solubility is not increased by the particle size reduction. So in this figure, the horizontal axis is the particle size in nanometer scale. The vertical axis is the relative solubility. Even when the particle size was reduced down to less than 200 nanometer, the relative solubility is increased only by 10%. And actually, these are really nice papers showing that the solubility is not affected by particle size, at least down to 200 nanometer. So when we are using the filtration for solubility measurement, we must be very careful about the interpretation of the data, because the pore size of a filter, which usually use, is about 0.2 micrometer, which is about 200 nanometer. So even when only a portion of drug particles pass through the filter membrane, it increases the drug concentration in the filtrate. It significantly increases the solubility, increase the drug concentration in the filtrate. So we must be very careful when we are interpreting the solubility data when we are using the filtration or centrifuge method. So this slide shows the mucus structure. So many people asked me, why microscale particles can penetrate into the unsaturated water layer, because there's a mucus layer. So I also looked into the literature about the structure of the mucus layer. And basically the pore size of the mucus layer is much greater than 1 micrometer. So basically even microscale particles can penetrate into the unsaturated water layer, like I mentioned, and like many papers suggested in the real experimental paper. So this is really a nice paper. Let's move on to the final part of my presentation. It's extrapolation to FA, considering the particle drifting effect. So this slide shows the structure of the human intestine membrane and also the microflux apparatus. So in humans, we have a plicae structure. This is the fold structure, and villi structure. On the other hand, in microflux, the membrane is flat without any this kind of fold and villi structure. So when we extrapolate the microflux data to human FA, we have to correct the effect of plicae structure and also the villi structure. And in addition, the thickness of unsaturated water layer may be also different. So we also correct the thickness of unsaturated water layer to predict the human FA from the microflux data. So this is a summary. So I proposed the particle drifting effect based on the theoretical FA equation and PEF equation, and also the real drug discovery and clinical study data. So the particle drifting effect, but at this moment, I'm not sure whether that particle drifting effect occurs by the self-diffusion, so Brownian movement, or by the convection, by the fluid flow. There's maybe two possibilities, and I'm really keen to learn which one is, occurring in vivo. And the microflux to FA prediction, considering the particle drifting effect, is now, implemented in the predictor from PION. So this is the results of from the PION with Maker University collaboration. And in that predictor, the effect of fold villi structure and unsaturated water layer thickness are corrected. So basically, we can now predict FA from the microflux data. So I'd like to thank the PION colleagues about the PION with Maker University collaboration. And this is a theme of the web discussion with PION scientists and our research group from this room. And then I also especially I'd like to thank my student, Shiori Ishii. She's a really good scientist, and she's now, working very hard to perform the microflux experiment. So thank you very much for your kind attention.
Thank you very much. Yes, I'm just checking the questions. I actually do have a question myself.
Yes.
So you have mentioned that this phenomena is something that can increase the in vivo absorption for solubility and stable water layer limited permeabilities. Can you give us any more guidance in terms of particle size or dose, or which are the more typical cases when this can have a bigger impact?
Okay, thank you very much. So we often observe the particle reducing effect when the particle size is less than 5 micrometer and the dose is over 200 mg in humans.
Okay, thank you. One more question.
Yes.
From an anonymous attendee. We have found the permeability of the smaller particles with a higher diffusion coefficients and higher energy is affected by temperature, as one would expect. Or have you found, yes, the permeability of smaller particles have higher energy and temperature has an effect?
Okay, that's really a good question because, temperature should have a significant impact on the Brownian movement. So I think that point can be, studied using the microflux. We can't change the body temperature, obviously. So, but we can study that point using the microflux. Thank you very much for your good suggestion.
Thank you. Another question. Does the fraction absorbed prediction by microflux only apply on non-supersaturating cases?
Well, that's another very good question. So actually, we are now measuring the flux from the solid dispersion, amorphous solid dispersion formulation, and try to predict the human FA for amorphous solid dispersion formulation. That's the point where we are working. Okay, that's a really good question.
Thank you. One more question, sorry, jumping all over the places where the questions are appearing. Is the unstable water layer changing based on the stirring conditions, or is a fixed number?
Yeah, that's really also a good question. So basically, it depends on the stirring strength. And I think the predictor has some equation to calculate the thickness of unstable water layer in the microflux system.
Thank you. Has this particle drift effect phenomenon been incorporated into gastroplasts?
I don't think so.
Yeah, this is a short question, short answer. Maybe a last
please
one.
ask gastropath, okay?
Yes.
I'm not. I don't know the whereabouts gastropath. Please ask them.
Yes, I don't know the answer to that question as well. What changes would you make to predict fraction absorbed percent across species, rat versus dog versus humans?
Yes, that's really a good question. So the first thing is about the dose strength. That's very important. So basically, we normalize the dose by the body weight, one thing, and also the surface area. So basically, the human intestine has a very huge surface area because we have the fold structure and villi structure. But in rat, the rat doesn't have the fold structure. And also in rats, the biomycel concentration is about 5 times higher than in humans. So we also have to correct the effect of biomycel. And in dogs, the pH is about 1 pH unit higher than that in humans. So basically, the intestinal pH is around 7 to 7.5 in dogs. In human, it's about 6.5. So we have to also correct for the pH. So these three are very important points to predict the species differences.
Thank you very much. We have 20 more questions. So we will try to answer those after the webinar. And now we will move on to the next presentation. Thank you very much, Kyo,
Okay, thank
for
you.
the great presentation.